In this video, we'll talk about restriction enzyme and restriction sites. So restriction enzymes are considered as molecular scissors because using restriction enzyme, you can cut DNA in specific location. So the restriction enzymes are mostly isolated from bacteria, but have you ever wondered that why restriction enzymes are required for bacteria? I mean, like, why does bacteria produce restriction enzymes? So this is a confusing question, right? And why does bacteria need to produce restriction enzyme? It turns out that the enemy of the bacteria, that means the bacteriophage, which kind of kill bacteria and survive on that, uh, they invade bacteria and put their genome inside. And the bacteriophage can be prevented from attacking E. coli if the bacteria is producing a restriction enzyme. So restriction enzymes work like a defense system for bacteria. So bacteria normally put their DNA inside. That DNA can integrate into the bacterial genome in a lysogenic cycle, or bacteria might also undergo a lytic cycle. So these enzymes, restriction enzymes, ensures that the virus, the phage virus, might not survive, and these restriction enzyme kind of cleave the DNA of the phage virus and thereby protects the bacteria against the phage virus infection. Now, that is why the restriction enzymes are a defense mechanism or immune mechanism for bacteria we can think of. Now, these restriction enzymes don't cleave their own DNA of the bacteria. Why is that so? Because soon after the DNA is replicated in the bacteria, it is methylated, and these restriction enzyme do not recognize DNA with a methylated at cytosine. So they don't cleave their own DNA, but they can cleave the foreign DNA that comes from the phage virus. And that is how restriction enzymes are useful for bacteria itself. But how that is important for biologists? And how do scientists even figure out this thing that bacteria has this kind of mechanism and we can use that mechanism in purpose of biology? So first of all, we should talk about the work of Brittany and Luria. By the way, the LB broth or LB agar that we use today to grow bacteria is named after them. So what they were doing, they were like working on lambda-phage, and they seen that lambda-phage can easily grow on E. coli plate, and they form plaques in the plate. And so when they changed the strain to a different E. coli strain known as E. coli strain K, they saw that there are few plaques produced. I mean, not compared to like E. coli strain C. They thought that there is something that is preventing the phage to infecting the bacteria, and thereby there are like less kind of bacterial viral plaque, and that might be strain specific. So they wondered what are the difference between the strain K and strain C. Later, they call them restriction factor because it restricts the virus to grow in presence of that strain. So there is some kind of factor, magical factor, that is preventing the viral growth, right? Later on, 10 years down the line, Arber and Michelson figured out that these protective factors or the restriction factors are nothing but enzymes which can chop off DNA and cleave the viral DNA. So that was a big discovery in the field. But problem is, they thought that can we try to use that restriction enzyme and try to manipulate the DNA or manipulate the genome of an organism. But only problem that they faced is like the restriction enzyme that they found was not specific. Now we know the restriction enzyme they found was a type 3 restriction enzyme which cut in a random location. But eventually, 10 years down the line, people found out a type 2 restriction enzyme. Type 2 restriction enzyme has specific recognition site, and they only cut in specific recognition site, not anywhere else. So they are very specific. So Thomas Kelly and his co-workers found that specific restriction enzyme subtypes, which they isolated from Haemophilus influenzae bacteria. Hind was the name of the first restriction enzyme, and they saw it cut into a specific sequence and specific location, and there are like specific length of fragments were produced from this cut. And that was a game changer in this field. All the field knew that they can use this strategy to manipulate genome and cut a specific length of DNA. And that is how the restriction enzyme came into the picture and molecular cloning started. Now, there are restriction enzymes which could produce sticky ends and which could produce blunt ends. It depends upon the choice of the restriction sites. For example, this is an example of a sticky end which is protruding out. In terms of cloning or ligation, this kind of sticky ends ligate better than the blunt end. Now, these are some examples of like popular restriction enzymes that are isolated from specific bacterial strain. And there are a variety of bacteria which produce different type of restriction enzymes. Nowadays, we use this restriction strategy and put these restriction sites into a cloning vector. For example, here it is plasmid. And in its multiple cloning site, we put the restriction recognition sites such that we can easily use these molecular scissors, which is these restriction enzymes, and cut that according to our own choice, and we can put our DNA of interest in that as well. So here is the workflow. So here is your plasmid, which has the restriction recognition site in the green zone, we can see, and we can cut that with restriction enzyme. And our gene of interest, we can also cleave it with the same restriction enzyme. Later on, we can ligate the two, and thereby our gene of interest is now inside in the plasmid of interest, and it is directed to the specific location where we want it, because there are multiple different restriction enzyme of our own choice, so we can use whichever combination we like to. And after that, we transfect that into the bacteria, and as the bacteria grows, our gene of interest is also getting increased in number in format of a plasmid. And that is a very important strategy, and that is how molecular cloning started. So restriction enzyme was a breakthrough in the process of molecular cloning.